Okay, this is chapter 17, which deals with the innate immune system. This is non-specific host defense. So with the immune system, we have different levels. We have essentially like 3 lines of defense. The first and second line of defense are very non-specific. This is going to happen whether it's E. coli or whether it's the flu virus. It doesn't matter, or even pollen. When we say it's non-specific, we just mean it doesn't matter what the foreign substance is. Your body just sees it as a foreign substance and it's going to attack it. There's a third line of defense that's known as the adaptive immune response, and that's what we will cover in the following chapter. It's not acquired over time. This is built in from day one, basically. It's an immediate response, so there's not a delay for things to build up. The whole idea is protection, and so it's trying to prevent pathogens from entering the body with the first line of defense. Don't let it penetrate. You can't cause infection if it can't even penetrate your defense system. If it does manage to penetrate through the skin or enter the body in some means, whether ingestion, inhalation, however, then the second line of defense is there to, you got past the first line, well, now we've got another means of trying to fight you. Excuse me. So this is going to involve several different things. It's going to involve some physical barriers. Mechanical defense is going to be involved. Your natural microorganisms, just the fact that they're there and taking up space, is going to play a factor. There are chemical defenses. Some of the chemicals and the enzymes in the body are going to play a huge role. Some of these are antimicrobial chemicals, so they're going to kill anything that comes in its way that's not you. There are proteins in the blood that are involved. There are cellular aspects to this, the granulocytes and the agranulocytes, which are divisions of your white blood cells, basically. We're going to look at all of these factors that are involved. So the physical defenses, several different things are involved here. You've got things like the fact that your cells in the skin are very tightly joined together and they're held together tightly. This, number one, helps hold your body together. Number two, it helps to prevent anything from penetrating through you. Your skin is a physical barrier. Your mucous membranes are a physical barrier. The endothelia is a physical barrier. So in this picture, it's showing you different types of the connections between cells. This may be a review for anyone that's had A&P, but you've got things like your tight junctions, your desmosomes, your gap junctions. Different means of holding adjacent cells together so they are once again held tightly right next to each other, and it's not like, well, we're supposed to be next to you, but I'm going to kind of float off a little bit, and then now there's a gap for you to get through. No, it's going to hold you tightly together. That prevents microorganisms from being able to penetrate. The skin is multi-layered. You have your epidermis on the outside, or the most superficial layer. Then right below that's the dermal layer. You have the hypodermis underneath it. On the surface of the cells up here on the epidermis, for those of you who've had A&P, you would remember up here, this upper layer of the epidermis, what's on the surface is dead. You're constantly making new cells and pushing up the dead ones basically to the surface, and the dead ones are being sloughed off. So if a foreign substance comes in and it lands on the surface of the skin, because you're sloughing off the cells, most likely it's just going to be sloughed right off. See you later. You're gone. You're out of here. That alone is going to help prevent things from being able to penetrate. In order for something to penetrate through the skin, it has to be able to get past those cells that are constantly being sloughed off. And so it's okay, can you manage to get past that? They're being sloughed off, pushing off, but you have to be able to push through. These cells are tightly held together, and you have to be able to get down through into the body What you do not want to have happen is for it to get down to the blood vessels. The epidermis does not have a blood supply. That's beneficial to us because once microorganisms, whether it's a bacteria, virus, fungus, whatever, once it gets in the blood, it can go anywhere in the body. And that becomes another whole huge problem to have to deal with. So if number one, the body, the first and second lines of defense are trying to keep things from entering the body. If they do, you immediately try to fight it, kill it, get it out. Don't allow it to penetrate even further into the body. Keep it contained. Don't let it get to blood vessels. Some cells, especially like along the respiratory system, are ciliated. All these fine little fibers, these are cilia. They're constantly moving. That helps to prevent foreign objects from entering as well. It's going to help move them, flush them away. The production of mucus. This is a goblet cell that's producing mucus. That mucus, like in the respiratory system, where you're producing the mucus and then we cough, and it helps move it out. It's helping to flush out the system. It's flushing all those foreign substances out, preventing them, once again, from penetrating further. So some of the mechanical defenses, you've got things like the shedding of the skin cells, like I had mentioned, coughing up the mucus or disposing of the mucus, expelling it, flushing of urine. That movement is going to flush out the urethra. That's going to help prevent things from getting established. Flushing of tears, like over the eyes. On the eyes, we'll see a bit, does contain an enzyme, the lysozyme, but every time you blink, you have the lacrimal gland that's producing the tears is above the eye over here, and so it's going to, every time you blink, tears are going to flush across the eye, and then they drain out the tear duct here. So that's helping to prevent anything from coming through the eye. Then, like I say, the kidneys, producing the urine, and then that urine is going to flush down the ureters. It's going to be stored in the bladder, and then it's going to be flushed out. That movement helps prevent, ideally, things from moving through or entering into the body. The microbiome, those are the resident microorganisms we've talked about before that are naturally on you and in you. A lot of people don't want to necessarily think about it, but if an area is already taken up, the space is already filled, then that means there's not available space for anyone else to come in. It's kind of like, if a parking lot, if all the parking spots are filled, it's full. You're not going to be able to fit another car in there because all the spots are filled. It's the same thing on the surface of your body and in different areas like the large intestines of the body. If all the niches are filled, there's no place else for anyone to come. And so there's only a limited amount of space and nutrients available. They're all competing for the same nutrients in the same space. So that's why, like when you wash your hands, some people mistakenly think, and we've talked about this in previous chapters, some of the terminology, do you want to sterilize your hands? No, because the method to sterilize is going to be way too harsh, and it's going to actually kill cells on your hands and cause more damage. You wouldn't survive it. But do you want to remove, when you wash your hands, do you want to remove all of the microorganisms on the surface of your hands? No, you don't. You want to, it's like, you want to get rid of the bad ones, but you want to leave the good guys there. That's kind of the way of visualizing or thinking of it. In terms of chemical defenses, there are a lot of chemicals and enzymes in bodily fluids, whether you're talking about tears, saliva, urine, whatever. Some of these are antimicrobial peptides, meaning that they will act against any microorganism. Some of them are protein mediators, some of them are cytokines, some of them are chemicals that are going to trigger or elicit the inflammatory response. So some of these chemical and enzymatic mediators in the sebum with the hair follicles, like say the saliva, mucus, gastric fluids in your stomach, intestinal fluids, urine, tears, cerebrum in the ear, the vaginal secretions. So in this example of a sebaceous gland, you've got your hair follicle here. You've got the sebaceous gland, which is producing various oils, etc. And it is going to help lubricate the hair. Number one, yes, the hair is coming up. There is an opening right here, which potentially microorganisms can come down. But if the sebaceous gland is producing chemicals that are helping to lubricate and protect the hair, the sebum, guess what? It's moving this way. It's moving out, so it's kind of helping to flush out, and it does contain chemicals that basically are bactericidal. It'll help to kill them. However, there are some that are able to get past them. Bactericidins are antimicrobial peptides that are found in the gastrointestinal tract. The GI tract what it will do is disrupt the membrane on bacteria. The skin, you've got chemicals that will do the same thing there. Defensins are throughout the body. Dermacidin, which is on the skin, interrupts and disrupts the membrane, and ion channels. So that's going to interfere with the functioning of the microorganism. And histatins in the oral cavity, in the mouth, all these are going to disrupt and end up interfering with the normal functioning of the microorganism, which is going to end up killing it. Plasma protein mediators, you have some that are produced in the liver, some that are secreted in the blood. Some of these are going to be secreted in response to the inflammatory response. They basically are going to inhibit or destroy the microorganisms. You have the complement system. Complement system, which we'll talk about this again with the secondary line of defense. And that plays a role again in the third line of defense, so we hear it a lot. The complement system is a group of proteins that become activated when microorganisms are present. So the initial presence of the microorganism, of the pathogen, is going to activate the protein. Now we call it a cascade effect because it's multiple proteins. So the first protein activates the second protein, which activates the third one, and it has to go in a very specific order. The bottom line is that it's going to target the microorganism for killing, basically. Ultimately, it's going to essentially punch holes in the cell wall and the membrane of it, and it's going to cause all the inside to leak out, which is going to kill the cell. So we'll discuss a little bit later. We're going to see again, talk about exactly how it works. But if you see complement, think, well, that signals the end of a microorganism because it's going to die. Cytokines, these are proteins that are used to help cells communicate, basically, with each other. It's going to give them different messages as far as do you need to start reproducing. Some of them play a role in chemotaxis, which chemotaxis refers to chemicals that are either going to attract other cells to it or repel them away. It's going to play a role with the inflammatory response as well. So this is just showing, excuse me, there's, with the complement system, there's 3 different ways of activating it. Bottom line is all of these right here that have a C and then a number, those are referring to different proteins. So they have to be activated in this certain order. The numbers, if you notice, not necessarily in order, they're numbered according to the order that they were discovered. Okay, so that's why if you look at it, why is 4 before 3? Yes, we know how to count. It's how it was discovered. So don't really worry about the differences on how it's activated. There are 3 different ways. Just once it is activated, it this final product is going essentially to attack the membrane of the foreign cell. As I said, cytokines are these messengers for communication between cells. There's also different ways that they can work. And then up here, interferons. Most people pronounce this as interferons. It's a type of cytokine. I was at a conference once, and some of us were surprised because different areas have different pronunciations. And the person giving the talk was calling them interferons, and it took the rest of us a while to figure out, like, what is he saying? So just be aware there are different pronunciations. Most people I know will say interferons. So what these are, they're signals, once again. When a cell is infected with a virus, it triggers the release, typically of a certain type of interferons. What do those interferons do? Some of them will go to other adjacent cells that are not infected with the virus and basically gives a signal to destroy the RNA, stop protein synthesis. Because if you do that, that's what the virus needs in order to replicate. So it's kind of like we're trying to do some preemptive work here. If the virus is released from the cell here and goes to the cell, if you have already destroyed the RNA, you need RNA in order to make proteins. So that means you'll never make proteins, which is what the virus needs to make its coat. So you're not going to make new viral particles. It's a way of preventing it from ever happening. Now, it also will trigger some of the neighboring cells, especially if they're already infected, to undergo this process, which is basically self-destruction. The cell is going to destroy itself. It also is going to activate some of your immune cells. Okay, we have an invader. You guys need to kind of step up your game here and fight this. In the blood, you have the liquid portions known as the plasma. And then you've got what's known as formed elements. That includes your erythrocytes, which are your red blood cells. Thrombocytes are your platelets. And then your leukocytes are your white blood cells. Your leukocytes are going to be subdivided into granulocytes versus agranulocytes. This involves your cellular defenses. The granulocytes, there's three types. There's your neutrophils, your eosinophils, and your basophils. The agranulocytes, you have your monocytes and your lymphocytes. And then your lymphocytes are subdivided even further into your natural killer cells or NK cells, your T cells, and your B cells. So all the formed elements, all the leukocytes, the erythrocytes, thrombocytes, they all start from an original stem cell, which then starts going through a process. They're all with the stem cell up here, and it goes through this process of differentiation, specializing into, it's kind of like getting a message of, okay, you're going to become a basophil. This chart is a summary chart describing the functions, how you stain for it, what's the relative numbers of these types of infections. So if we look at them, the granulocytes, when they get that name because when you stain them, it looks like somebody dumped a bunch of sand inside the cell that has all these granules in them. So that's how it gets its name. The neutrophils, when you stain it, they look pale, light purple, kind of a lilac granules. What do neutrophils do? They basically fight against bacteria. All of the leukocytes, all the white blood cells have a nucleus in them, but the shape of them is different, and you can use that to help identify it as well. So not only by looking at the color of the granules after you've stained them, but look at the shape of the nucleus. So a neutrophil has 3 to 5 lobes on the nucleus. Like I said, we know what the relative numbers of these are going to be. if you had a bacterial infection, you would expect the neutrophil numbers to be really high. The eosinophils, these are kind of a reddish orange in terms of the color of the granules. They have 2 lobes on the nucleus. They are responsible for phagocytizing or eating, killing the protozoans and the helminths. They also, the numbers tend to go up when you have an allergic reaction. The basophils have a dark blue, dark purple granules. They have 2 lobes to the nucleus. The basophils, those numbers go up when you're having an allergic reaction. Mast cells are similar, but mast cells will leave the blood and then are found in the surrounding tissue. The basophils are going to stay in the blood. Something like that, we know what the normal range in terms of numbers that these should be in. Now, one thing they don't always tell you that I often mention, and this is using your critical thinking skills. And here's an example of how you can use the information that's just on this page to tell you something. Years ago, after I had my second child, I developed a really bad bacterial infection. So they started me on medication. Then I got a whole lot worse. And I mean a whole lot worse. So I just started the medication. Well, if any of you have been sick and you start antibiotics, you know that sometimes it takes, a few days of taking the antibiotics before you kind of get ahead of the infection and then start feeling better. So yeah, when you first start the medicine, oftentimes you're still feeling obviously sick. And sometimes you might feel a little bit worse before you start feeling better. However, because I suddenly started feeling so much worse, my concern was I was not sure if I was having an allergic reaction to the medication they put me on. And so in this situation, here I was, sick, suddenly getting much sicker. I have a newborn and I have a 2.5-year-old. And with my luck, it's Christmas Eve. And so I finally told my husband, you have to take me in. Something is wrong. We've got to figure out, if I'm having an allergic reaction to the medication, I need to stop this medicine, because if it's an allergic reaction, it's just going to get worse. And then they need to switch medication on something that will work so I can get better. If I'm not having an allergic reaction, okay, then I need to know that it's fine to continue taking the medicine that was prescribed. So at that time where we were living, there was not a hospital in town because we lived outside of Albuquerque. Albuquerque had the hospitals, not Rio Rancho. So we had an urgent care. So we went to the urgent care. Like I said, it's Christmas Eve. It's like, we're one of those annoying people where it was like 5 minutes before closing, night of a holiday, and we come in, and you could see the look on everyone's faces like, oh my goodness, really, are you kidding me? Go lock the doors or something. So we're coming in, and to make matters worse, we had no family in the state at all, and so we had to bring the kids with us. So here we come with a newborn and a 2.5-year-old coming in, and they're looking at us like, okay, who's the patient? And it's me. Meanwhile, my 2.5-year-old is crying because, where's Santa gonna find us? And I want to be at home and all this. So, like I say, the looks on their faces were just like, oh goodness, we thought we were getting home early. And so I told them, I don't normally tell people in that type of situation what I do, because I find it usually makes them really nervous, so I don't tell them. Anyway, I go in there and say, okay, look, we already know that I have an infection. They've already run tests. They know it's a bacterial infection. What my concern is, because I'm feeling worse, is am I having an allergic reaction to the medication? And here's what you can do. Stick my finger, take a drop of blood, and go do what is known as a differential blood count. A complete blood count is where you just count the number of leukocytes, the number of erythrocytes. Fine, you have the total number. A differential blood count is where they look at the leukocytes And remember, you've got these 5 types, and they distinguish and do the ratio of what percentage are neutrophils, what percentage are the eosinophils, basophils, etc. So you know what the normal ratio should be, the normal numbers. And then I told him, I said, we already know that I have a bacterial infection, so you are going to expect the neutrophil numbers to be sky high. We know that because I have a bacterial infection. What you need to look at, don't even bother looking at the neutrophils. Don't count them because you know they're going to be high. We're not worried about the lymphocytes. We're not worried about the monocytes. What we want to look at right now are the eosinophils and the basophils, because both of those respond to allergic reactions. If those two are within normal ranges, then that means I'm not having an allergic reaction. I can keep on the medicine I'm at. It just needs more time to kind of get ahead of this infection because it's so bad. If the eosinophils and or basophil numbers are really high, then that tells us I am having an allergic reaction and we need to change the medication that I'm on. You can do this in 5 minutes. And that's when they look at me and they're like, what do you do? How do you know this? Oh, and then I usually get the, oh, I hated microbiology when I took that class. And I'm like, okay, I won't take it personally. That's fine. Just please go look at the blood sample. If you want, I'll look at the blood sample. Just please go do it. So they did that, and they came back in about 5 minutes, and they were kind of like, oh, no one's ever really talked about this. And that's something that they often don't teach you in terms of, say, nursing skills. But like I say, it's just have those critical thinking skills. If you know what the cells do, and if the numbers are abnormal, why? Well, let's think about that for a little bit. And so in my case, they came back and they said, well, your neutrophil numbers are really high. And I'm like, yes, we know that. That makes sense. What about the eosinophil or basophils? And they said, those are in normal range. Everything else is normal range. It's the neutrophils that are really high. And I said, okay, that tells me that, yes, obviously we know I have a bacterial infection. This tells me I am not having an allergic reaction, so we can all go home and I continue taking the medicine. You don't have to change it. It just needs a little bit more time to catch up. And they're like, oh, okay. So now everybody gets to go. You guys can lock the door as I leave. My 2.5-year-old was happy that we were going home and Santa Claus found her, and all of that. But it's using those critical thinking skills. These granulocytes attack different things. You know what the normal concentration in a healthy individual who has no infection, what those numbers should be. And so this will tell you that, yeah, there's an infection going on. Recently, I was dealing with my father, who's 92 and was in the hospital. We found out he had COVID, and then we were concerned about him developing pneumonia. And so they were running some numbers, and they're like, some of these numbers, when my siblings were getting the lab reports going, oh, some of these are really high. It's like, yeah. And they said, well, the doctor said that indicates an infection. Yes, it does. Yes, it does. And then we were slowly watching daily as those numbers came down. It's like, okay, and somebody had asked me, what does that mean? Is that good? And I'm going, yes, it is. It means that we're getting a handle on the infection. They're controlling it, preventing it from spreading and getting worse. So this is showing you a schematic drawing versus an actual picture on the top row of these 3 different types of granulocytes. So you've got your neutrophils, the light purple. Like I said, it has multi-lobes. 1, 2, 3, 4. It's not what you would consider like your normal looking nucleus. Eosinophil, which is the red-orange color, 2 lobes here. And then the basophil that has the purple, dark blue, dark purple. Mast cells are similar to the basophils, however, they're going to be out in the tissue. So, Agranulocytes, these are the monocytes and the lymphocytes. Monocytes have a very large nucleus. It's almost, if you look at the cell, it looks like the whole thing is nothing but the nucleus. And this, I know, can be a little frustrating for students. When these cells are in the blood system, so in the blood vessels, they're known as monocytes. When it leaves the blood and is traveling in the interstitial space, the space between the cells of the tissues, outside of the blood, we know, or we call it as a macrophage. Yes, it's the same type cell, but the name just distinguishes the location of it for us. Also, these are dendritic cells that are found in the skin and in the mucous membrane. Lymphocytes, most people think of them with the T cells and the B cells that are involved with the adaptive immune system, which we cover next. It also includes the natural killer cells, or the NK cells. Natural killer cells can recognize and kill abnormal looking cells or cells that are infected, especially with viruses. Just so you know, there is a lot of research going on right now with natural killer cells, trying to see if they can recognize abnormal cells. Oftentimes this is because, like, when a virus infects a cell, it puts a signal on the outside of the cell so it can recognize, oh, that's not supposed to be there. Where are some of the research is looking at, can we use the natural killer cells, if they can recognize abnormal cells, can we get them to recognize cancerous cells? That gets tricky because cancerous cells are technically our cells who have lost some of the regulatory controls for growth. So basically they're growing uncontrollably. And so it's kind of a fine line there with the natural killer cells. Obviously, as the name implies, the purpose is to kill cells. So can we somehow get them to recognize our abnormal cells, leave our normal ones alone, but kill the cancerous cells? So this is showing the natural killer cells here. Like I say, we're trying to figure out, there has been some success. It's not necessarily widespread, but there is some success with using the natural killer cells. The monocytes, as I said, look at the nucleus. See how, I mean, it's almost a whole cell here. In this sample, it's in the blood. For those of you who have not had anatomy, remember the red blood cells do not have a nucleus. So if it has a nucleus in here, you can see that it's going to be one of the leukocytes. And then in the tissue sample, there are the macrophages. Large nucleus still. Phagocytosis, this is the process of where you're attracted to pathogens and you're going to be able to recognize it as a foreign substance, and you will ingest it and you will destroy it. Years ago, I used to describe it as kind of like Pac-Man, but as times change, and most of you probably don't know what that is, pick your favorite little computer game, and it's like seeking out and destroying the enemy. Injured cells secrete certain chemicals when they get injured that will attract the phagocytes to them by what was known as chemotaxis, specifically positive chemotaxis. Remember I said chemotaxis either attracts or repels. When an injured cell, when the injury first occurs, it secretes these chemicals that it's a positive chemotaxis. It attracts the phagocytes to them. Phagocytes will also, in addition to when they see the pathogens, ingest them and kill them, they will also ingest damaged cells. They will ingest like tissue debris. When you've had an injury and damaged some of the tissues, it's a cleanup crew. So it's not only pathogens, but any damage and debris it's going to phagocytize. The phagocytes are going to come in and ingest that. And so this, in this drawing, is showing you've got an injury site here on the skin. The cells that were injured they are going to release these chemicals that are they're known as cytokines. Cytokines are going to trigger the inflammatory response, and they are going to attract some of these, in this case, the monocytes, which become the macrophages, which carry out phagocytosis. And so the cytokines are released into the tissue, they make it into the bloodstream, and oh, here we go. It's going to start attracting. Here are some of your cells that are going to now carry out phagocytosis. They're going to clean up the debris. They're going to start attacking those foreign invaders. When a cell engulfs through phagocytosis, when it engulfs a foreign substance, such as this bacteria here, it's going to basically enclose it in a vesicle. And then remember the lysosomes that we have? That is going to fuse with it right here. And if you remember, the lysosome has highly digestive enzymes. So it's going to break down. That's why we call it the recycling center. It's going to break down that bacteria, completely destroy it. What happens to the components of the bacteria? This is where, remember when I made the comment about the unity and diversity of life a couple chapters ago? When it breaks down the bacteria, and it's breaking down the proteins that are in there. Proteins are made up of amino acids. But the amino acids are the same between all cells. So when it breaks down the amino acids and it starts to release it into the cell, let's say it's releasing, well, it's going to be a whole variety of amino acids. Glycine, leucine, alanine, all of those amino acids. Your cell can use those. When it breaks down the DNA of the bacteria, your cell can use the deoxyribose sugar. It can use the phosphate. It can use the adenine, the thymine, the cytosine, the guanine. So the cell is going to use whatever it can. Whatever remains that it cannot use, then it will package that waste and it's going to get rid of it. But the cells, any type of cell, is very good at recycling. Inflammation. There's two types of inflammation. There's acute versus chronic inflammation. Acute inflammation usually is good. It's short-lived. It's very localized. It's good from this standpoint. We'll see in a moment when we talk about the phases or steps of it. It's going to be a good immediate response to try to help prevent infection. Chronic inflammation, this is long-term. This is not so good. It's kind of like, initially inflammation was good, but you need to stop it because if it continues for long-term, it can lead to scarring. It can lead to formation of different granules, and it becomes more harmful in the long term. So what are the signs of inflammation? Redness, which is known as erythema, swelling, which is edema, heat, pain, and sometimes altered function. Why do these happen? Because when you have an injury, let's say you cut your hand, what is happening? And this all ties into the innate immune response. As soon as you cut yourself, those damaged cells release those chemicals, the cytokines. They're releasing those chemicals. Those are the ones we just talked about, that through positive chemotaxis are going to attract the phagocytes to them to try to clean up. It's going to attract the macrophages. It's going to trigger the monocytes to move out of the blood vessels, and then they become the macrophages. Sometimes collectively we just refer to them as phagocytes because they carry out phagocytosis. This all starts the process. So once again, you cut yourself. You've damaged some of the cells. The cells release the cytokines. The cytokines are attracting and they start the inflammation process. Basically what it's saying is, hey guys, we got a problem over here. We have a breach in the integrity of our defense system. I mean, it is kind of like military, if you will. You know, the integrity of the skin has been breached. We got a breach in the outer barrier. We need help. We need to contain. We got invaders in here. Get here now. And so those cytokines, they end up in the blood system, which, as I just said, they're going to attract the fighters. First line of defense. Here come the monocytes. They leave the blood vessels, so now we call them the macrophages. They are going to come to this area. The blood vessels in the area right near where the injury has occurred, they become more permeable or leakier. For fluids, this enables and increases the chances of these helpful cells, the monocytes, to be able to leave the blood vessels. And then we change the name to the macrophages, just to annoy you students. But they leave the blood vessels. Why? Because they're responding to the cytokines that have been released, saying, we have a breach, we have a breach, we need help. Send reinforcements in. So here come the reinforcements. They come in. Well, it's not just the macrophages that are able to leave the blood vessel easier. Because they're leakier, you have more fluid. Because you have fluid that's leaking out, that's where the swelling comes from. The blood vessel is actually going to increase in diameter in response to the cytokines so that you get more cells. You get more blood to that area, meaning you get more cells. Because you've increased the diameter, you're increasing the blood flow to that area. That causes the redness. Blood carries heat with it. So if you increase the blood flow to that area, not only does it turn red, but it's going to feel warm. That's where the heat's coming from. Because you have this increased blood flow, you have this increased fluid that's leading towards the swelling. What is the swelling doing? It's pushing against the nerves in the skin. If you've had A&P, well, everybody, even if you haven't had A&P, we're all aware there's a lot of nerves in the skin, and you push against it, it's going to hurt. So it's all this extra fluid, there's only so much space, it's pushing against the nerves, that's what's causing the pain. And depending on how long this lasts, this could alter the function in that area. Okay, so here's a slide that shows the inflammatory response. You have an injury right here, a cut, as you can see. All the green here, these are the bacteria. These are the invaders. So you have this break. The damaged cells release the cytokines that end up going into the blood vessel, saying, we got a problem here, guys. We got a break in the integrity of the skin. The perimeter has been breached. We need help. Send in reinforcements. Part of that response is going to be increase the diameter, dilate the blood vessels. That's increasing the diameter of these blood vessels. That increases the blood flow to increase the chances of bringing in your reinforcements. The cytokines that are released saying, hey, we got a problem. Here's where we have a break. That is attracting these cells that are coming in. That their purpose is to carry out phagocytosis. So the blood vessels become leakier to allow more fluid to come in. It's allowing these cells to come out. They are going to the site of where you've got the damage. How do they know to get there? Because they're being attracted by those cytokines. Remember that positive chemotaxis, that's what's attracting them there. It's saying this is where we need the help. You've got the increased fluid coming out that leads to the swelling. You've got increased blood flow to the area that leads to the redness and to the warmth. And this excess fluid that's coming out, as well as the cells, is pushing against the nerves that's causing the pain. And what is occurring here is that these cells are coming out. You want to seal off this area so that the invaders cannot penetrate deeper. You want to contain it, seal it off and contain it. This green bacteria right here, as you can see, it's about to be toast. It's being eaten by that phagocyte. You've had better days. Seal it off. If these bacteria are able to get past and into the blood system, we have a major problem now because it can go anywhere in the body. And now treatment becomes a whole lot harder. So you want to isolate and contain where this breach occurred. And so that's why you have this whole communication system. We have an injury. We have a breach in the perimeter. I need reinforcements. Send them in now. Okay, increase the blood flow. Here come the reinforcements. They're coming in. They're going to seal off this area. They are going to start attacking and destroying any foreign substance, the pathogens. In this case, the bacteria that are there. Kill them off. Don't let them get any, deeper into the body. Now, the phagocytes are also, once they have taken care of all the invaders, like I say, they're also the cleanup crew. They are going to be engulfing any debris in this area, and that starts the healing process as well. Chronic inflammation is good. This is good on a short-term basis. It's protecting yourself. It's preventing this invasion from becoming worse. You can very easily deal with an infection in a very small area. The body can deal with that. If it becomes systemic, where it's in the blood and goes everywhere in the body, that's a whole new ball game. You don't want to go there. So acute inflammation, just for the short term, that's good. That is the body's response. It's doing what it's supposed to do to respond to basically a foreign invader. Chronic inflammation, because it is long term, short term you're okay with this. Long term, that starts adding in additional problems. That's not good. It can lead to scarring, as we said earlier. So acute inflammation is good, chronic inflammation is not. This is the way the body is responding overall for the innate immune system. It's a combination of physical and chemical means. You have your first line of defense, which is preventing the organism from even entering the body. It's a back and forth. And I do often describe it kind of like war games or chess games or whatever. It's as though, okay, the body has means of keeping you out. The first line of defense, well, we've got this nice intact skin. And it's very hard for organisms to penetrate through the skin. So it's kind of like, once again, we're looking at things from two different perspectives. From our perspective as humans, we're like, ha, I've got this nice layer of skin that is going to prevent you from getting in. It's a nice barrier. And then the bacteria is like, ha, oh, you thought that was going to stop me? Well, you know what? I make some enzymes like collagenase, hyaluronidase, where I can penetrate through the layer of skin. I'm coming in. And they're like, oh, so you got past my first line of defense. You thought you were in. You thought you won. Well, guess what? I've got some other tricks up my sleeve because I have my second line of defense, which includes the inflammatory response. So you thought that through this cut you were going to be able to enter into the body. Surprise! You tried to enter. We've got things like the complement system, which is going to kill you. We've got the inflammation system, which is going to attract cells that are going to kill you. And so just because you got past my first line of defense, you thought that was it? Well, I've got a second line of defense. As you're going to find out, the microorganisms, such as the bacteria, sometimes they kind of come back with, oh, you thought that was going to stop me? Well, I've got some tricks up my sleeve too, so guess what I can do? And then for humans, what we do is then we say, oh, well, did I forget to tell you that I have not only a first and second line of defense, but I also have a third line of defense. That's the adaptive immune response, and that's going to be very, very specific, as we'll see in the next chapter. First and second line defense, the innate immune system, is non-specific. This response, whether it's making complement or whether it's the inflammatory response we just talked about, this will occur whether it's a bacteria, whether it's a virus, whether it's tree pollen. It responds just to the fact that it is something that is not you. It doesn't matter what it is. It's not you. In the next chapter with the adaptive immune response, we're going to see now it is very specific. It recognizes the difference that, oh, this is the H1N2 influenza virus. And the response, I can recognize that. And I will distinguish between the COVID virus. I will distinguish between the measles virus. I can distinguish between E. coli versus salmonella. It's very, very specific. All of this is to protect us, and it's a response to foreign invaders.